Welcome to today's lesson on genetic engineering. In this lesson, we will explore the process of modifying the genome of an organism by introducing a gene from another organism. In today's lesson, we will start by understanding what genetic engineering is and look at the specific steps involved in the process, such as selecting the target gene, inserting it into the host organism, and ensuring it gets integrated properly. We will examine some real-world applications of genetic engineering, such as in medicine and agriculture. Finally, we will discuss some advantages and disadvantages to the process of genetic engineering. So far, you have had lessons on genetic variation and selective breeding. These topics have provided you with a good understanding of how genetics can influence the characteristics of living organisms, and how humans have traditionally selected for certain traits in plants and animals through breeding. Building on this foundation, genetic engineering allows us to precisely introduce new traits that might not have been possible through traditional breeding methods. This cutting-edge technology offers the potential to solve complex problems, improve crop yields, create medicinal treatments, and even address environmental challenges. Let's get started. To begin, let's learn what genetic engineering is. Genetic engineering is the process of altering the genetic makeup of an organism by introducing a gene or genes from another organism to achieve a desired characteristic. This approach allows scientists to manipulate genetic material to achieve specific outcomes, such as disease resistance in crops or the production of human insulin by bacteria. The process of genetic engineering involves identifying the desired gene from an organism and cutting that gene out of its DNA. This DNA is then transferred into the cells of another organism. Organisms that have had their genes modified in this way are known as genetically modified organisms, or GM organisms. Genetic engineering has a wide range of applications in different fields. In medicine, one of the most well-known applications is the production of human insulin by genetically modified bacteria. Before genetic engineering, insulin needed to be extracted from animals, which was less efficient and could cause allergic reactions in some patients. Bacteria has been genetically modified so that it can produce insulin in large quantities. In agriculture, crops have been genetically modified to improve yield, resist diseases, and produce bigger fruits. For example, GM crops like golden rice are engineered to contain beta-carotene to combat vitamin A deficiency. Genetic modification is also used to make animals produce medicinal substances. For instance, certain sheep have been engineered to produce proteins needed for treating certain conditions in their milk. Gene therapy is another use of genetic engineering. Scientists are exploring genetic modification to treat inherited diseases by inserting functional genes into affected individuals. This approach offers hope for curing genetic disorders that currently have no effective treatments. There are several advantages to using genetic engineering. Firstly, increased yield. GM crops can produce more food, which can help address hunger in developing countries. There can also be nutritional benefits, as genetic engineering allows crops to be fortified with essential nutrients, improving overall health. In medical treatment, the production of crucial drugs and potential cures for genetic disorders can be life-changing for many patients. Despite its advantages, genetic engineering also has some disadvantages. There are health risks due to uncertainties about the long-term effects of consuming GM foods, including potential allergies. There can also be environmental impacts, as GM crops may reduce biodiversity by affecting wild plant and insect populations. There is also a risk of gene transfer. There is a risk that modified genes could escape into the wild. For example, GM crops that are resistant to herbicides could potentially interbreed with weeds, creating superweeds resistant to herbicides. Genetic engineering is a field with significant potential, but also substantial controversy. Ethical considerations, potential health risks, and environmental impacts are all points of concern. The debate continues as the technology evolves and its applications expand. Decisions about the use of genetic engineering need to balance the benefits with the possible risks, considering both immediate and long-term consequences. In today's lesson, we focused on the concept of genetic engineering, which involves modifying the genome of an organism by introducing a gene from another organism to confer a desired characteristic. We then explored the basic principles of genetic engineering, including the process of identifying and isolating a desirable gene. This gene is then incorporated into the target organism. We also examined some practical applications of genetic engineering, such as in agriculture, where crops are engineered to be resistant to pests or to tolerate herbicides, and in medicine, where it is used to produce insulin for diabetes treatment and gene therapy. Additionally, we looked into the potential benefits and ethical considerations of genetic engineering, including how it can lead to improved yields, disease resistance, and medical advancements, but also raises questions about environmental impact, food safety, and moral implications. By understanding genetic engineering, you now have the knowledge to appreciate the mechanisms behind these technologies and their significance in scientific and everyday contexts. This knowledge will be crucial as we continue to explore more advanced topics in genetics, variation, and evolution.